Welcome back to the deep dive. Our mission today is, it's pretty crucial. We're tackling a massive stack of cutting-edge research here. We really want to understand this fundamental shift happening right now in biopharma. It's all being driven by generative AI and these large foundation models. And look, this isn't just about tweaking existing drugs. It feels much bigger than that. It seems like a potential revolution aiming to completely redesign the whole process, from finding that very first lead molecule all the way to running the final clinical trial successfully.
Yeah, absolutely. And if you're trying to figure out where the next exponential growth spurt is coming from in health sciences, this is definitely the area to watch. The context here is just so important to grasp. I mean, the traditional way of developing drugs, it's frankly catastrophically inefficient. We're talking about a journey where the cost to bring just one single novel drug to market usually blasts past 2.8 billion US dollars. That's billion with a B. And the timeline, if you count the initial research phase, it often stretches out beyond 12 years. It's incredible. And the worst part, most candidates just fail somewhere along that long road. That staggering inefficiency is precisely why this deep dive matters so much right now.
That cost and that timeline, I mean, it's just eye-watering. That's the historical bottleneck, isn't it? The big wall that maybe, just maybe, generative AI is finally equipped to start breaking down.
Precisely. You nailed it. Gene AI really represents a definitive paradigm shift. It's a whole new way of thinking. Instead of the industry just doing passive virtual screening, which is basically filtering through huge databases of chemicals we already know about, we're moving toward active, intelligent molecular generation. The models are actually designing completely novel chemistry, sometimes from first principles. The big goal here is to compress those multi-year timelines, which are so heavily weighted by just endless trial and error chemistry experiments, down to maybe months of focused computational design. So for you, the listener, the goal today is really to map out the specific AI architectures that are making this huge change happen. We're talking everything from equivariant graph networks to diffusion models, and crucially to understand the immediate translational impact they're having across different areas. Small molecules, yeah, but also complex biologics like proteins and antibodies, and even, surprisingly, the operational side of running clinical trials.
Okay, let's really start by digging into the sheer scale of the problem AI is trying to tackle here. When we talk about chemical space, all the theoretically possible compounds out there that might make a decent drug, the numbers just get, well, astronomical isn't even strong enough.
They really do. It's hard to wrap your head around. The theoretical chemical space is estimated to contain somewhere between $10 around and $10 potential drug-like compounds. I mean, just to put that in perspective, $10 is roughly the number of molecules in, like a few drops of water. So $10 is just incomprehensible. Traditional empirical screening, even the fancy high-throughput methods we have, can only ever physically test a tiny, tiny infinitesimal fraction of that chemical universe. It's genuinely a situation where brute force completely and utterly fails. You just can't test everything.
Yeah, it sounds fundamentally intractable for human chemists working alone or even in large teams. It's just too big.
Yeah.
Yet, we're seeing this rapid adoption of AI-driven methods. It feels like the industry is really seizing this opportunity. What kind of commercial acceleration are we actually seeing on the ground?
Oh, the acceleration is already quite measurable. It's not just hype. Right now, you've got these AI-focused biotech companies, and they have over 150 small molecule drugs currently in the discovery phase, and maybe even more impressively, 15 that have already progressed into human clinical trials. That's a significant number moving forward. But what's really fascinating here, and honestly what's driving some of these frankly massive valuations you see in the AI drug discovery sector, think about NVIDIA's market cap compared to the entire biopharma industry, it's getting comparable, is that this AI-fueled discovery process is expanding by almost 40% each year. That's the growth rate.
Wow, 40% annual growth. That's an enormous pace of adoption. But let me just push back a little on the optimism for a second. Is this growth really driven purely by solid scientific breakthroughs? Or is there maybe a significant chunk that's fueled by, venture capital enthusiasm and maybe a bit of hype around AI?
That's a really critical question to ask. And yeah, look, there's always an element of hype around any frontier technology, let's be honest. But this growth genuinely seems underpinned by real technological leaps, particularly in the ability to generate novel, high-quality candidates. The market, the investors, they recognize the potential here to turn those billions in R&D costs and those decades of painful trial and error into highly efficient computation. It's a numbers game when you see companies actually succeeding in generating novel, patentable chemistry in like a matter of weeks and then rapidly getting those candidates into preclinical testing. Well, the efficiency game is kind of undeniable. That core business case slashing late-stage failure rates by designing safer, more effective drugs right from the very beginning, that's the strongest driver behind that 40% growth number, I think. It's about de-risking the whole pipeline.
Okay, that makes sense. So let's really nail down the definition then. What exactly makes generative AI generative in this specific chemical context? How is it different from the older predictive AI models people might be more familiar with?
Right. So at its heart, generative AI works through statistical modeling and iterative training, yeah, but the key difference is its output. It creates something new. Unlike predictive AI, which might say, classify whether a known molecule is likely to be toxic or not, Gen AI actually generates new data samples. These new samples statistically resemble the input data, meaning they look like valid, stable chemical structures, but crucially, they haven't necessarily existed before. They are novel creations. In the chemistry world, the core idea is using these machine-learned chemical grammars, basically. The probabilistic rules of how atoms like to bond, how they interact, what kinds of functional groups are stable and useful to intelligently construct entirely new drug candidates, almost from scratch.
Right, building blocks and rules. And this naturally brings us to foundation models, or FMs. They're reshaping pretty much every industry right now. We're all getting used to large language models, LLMs, ChatGPT and the like. But how do these big foundation models apply to something as different as DNA, RNA, and protein sequences? It seems like a leap.
It does seem like a leap, but the core principles are surprisingly similar. FMs are really defined by their ability to learn from absolutely massive amounts of unlabeled data first, and then critically, they generalize that learning. They can excel in a whole diverse range of downstream tasks after just a bit of specific fine-tuning. Their emergence, why they're working now, is really down to three key factors converging. First, the sheer availability of massive, organized, but crucially unlabeled biological data think sequence databases. Second, our technical ability to just keep increasing the model size, making them bigger and bigger. More parameters seem to help. And third, this famous scaling law of generalizability. Basically, the observation that the bigger the model and the bigger the data set it learns from, the better and broader its performance becomes across many tasks.
Okay, so massive data, massive models, and this scaling effect. But how does that conceptual framework really translate into the nitty-gritty of biology? Are we seriously saying protein sequences are just strings of letters, kind of analogous to human language?
Conceptually, yes, that's exactly the viewpoint that's proving so powerful. In computational biology, we've started viewing DNA, RNA, and protein sequences, those long chains of amino acids or nucleotides, as a form of natural language. Think about it. A protein sequence is essentially a set of complex instructions. It dictates precisely how a highly complex 3D structure will fold up and ultimately how it will function in the cell. Once you adopt this kind of linguistic viewpoint, you suddenly find you can successfully apply the very same underlying transformer architectures that power the big LLMs, specifically things like BERT and its variants, to interpret and even generate these biological instructions. This allows us to do amazing things, like predict how stable a protein fold will be, or even guess its function just by reading the sequence. It's like the AI is learning to speak, read, and now even write the fundamental language of life. All right, so to really appreciate the depth of this revolution, we kind of need to peek under the hood. Let's look at the crucial model architectures that are actually underpinning Gen AI in drug discovery, and importantly, the different kinds of data representations they have to handle, because it's not all the same. We're talking 1D strings, 2D graphs, complex 3D geometry.
Okay, sounds good. Let's start with some of the maybe foundational approaches, like variational autoencoders or VAEs. What made them initially appealing for designing molecules?
VAEs were, and still are, quite popular because they have this sort of elegance in how they handle complex data distributions. Essentially, they function by taking a complex input, like a molecular structure, and mapping it down into a continuous, lower-dimensional latent space. You can kind of imagine it like a simplified map where molecules with similar properties end up being grouped close together on the map. And this mapping allows for something really useful, property-guided interpolation. So if you find two different molecules, let's call them A and B, that both have some desirable properties and they're at different spots on your latent map, you can actually sample a point exactly halfway between them on that map. Then you decode that point back into molecular space, and boom, you generate a totally novel molecule, C, that ideally blends the beneficial features of both A and B. It's like guided creativity.
That interpolation idea is neat. And you mentioned a success metric earlier involving really impressive precision in the 3D structures they generate.
That's right. That's where the advancements have come in. Basic VAEs have evolved into more sophisticated conditional VAEs, or CVAEs. These CVAE frameworks can actually integrate specific 3D constraints during generation. For example, you can feed it the known shape of a protein's binding pocket, and the CVAE will then generate molecules designed specifically to fit that pocket, often achieving remarkably low root mean square deviation, or RMSD. We're seeing numbers often less than 1.5 angstroms from the target pocket shape. Now, 1.5 angstroms, that means the generated structure fits the target lock almost perfectly. It's very high geometric precision.
Okay, that is high precision. But you also mentioned some trade-offs like this thing called mode collapse. What exactly is that? Does it mean the AI just gets stuck in a rut, like it keeps designing the same 4 or 5 molecules over and over, even though chemical space is supposed to be enormous?
That's a perfect analogy, actually. Mode collapse is a really significant limitation for many generative models, not just VAEs. It's exactly what you described. The generator finds a few easy wins, a few structures that score well according to its objective function, representing just a few modes of the overall data distribution, and it basically stops exploring the rest of the vast chemical landscape. So it fails to generate real diversity, which kind of defeats the whole purpose if you're searching for genuinely novel therapeutics. You don't want just minor variations on known themes. This issue, along with sometimes significant computational complexity, definitely pushed researchers to explore alternative architectures.
Okay, which naturally leads us to generative adversarial networks. They have a different mechanism, right? More competitive.
Yeah, GANs operate very differently. They utilize this fascinating zero-sum game mechanism. You have two neural networks, a generator that tries to create synthetic molecular structures, and a discriminator that tries to tell whether a given structure is real from a known data set or fake, created by the generator. They are adversaries. They compete directly. The generator tries to fool the discriminator, and the discriminator tries to get better at spotting fakes. As they compete, both networks typically improve. This competition often leads to the generation of very high-quality, realistic-looking molecular structures or images. That high quality is definitely their main strength. But, and it's a big but, GANs are notoriously difficult to train effectively. They are very prone to that same mode collapse issue we just discussed, and also to significant training instability. The whole training process can just diverge or oscillate wildly, making them somewhat unpredictable in a reliable drug design pipeline.
Okay, so VAEs have elegance, but maybe mode collapse issues. GANs have quality, but training headaches. It sounds like the field needed something else. And the real shift, especially in the last few years, seems to have been towards diffusion models. These seem to be popping up everywhere, especially for high-fidelity 3D generation tasks. What's the core conceptual difference? What gives them the edge?
Right, diffusion models have really taken center stage recently. They managed to avoid many of the stability and training problems that plagued GANs by fundamentally changing the generation process itself. They typically use a two-phase approach. First, there's a forward pass where they take real data, like a molecule structure, and systematically, step-by-step, inject random stochastic noise until the original data is completely obscured, basically turned into pure noise. Then comes the crucial part, the backward or reverse pass. The model learns to reverse that noising process. It learns to take the pure noise and iteratively, step-by-step, denoise it, gradually reconstructing the original structured data. It's a much more controlled, mathematically stable process of, you could say, structured destruction, followed by learned restoration.
That sounds quite elegant, actually. A controlled way to ensure the final structure is faithful to the learned data distribution. But okay, to make that work reliably in three-dimensional space, where orientation matters, you need to ensure the generated molecule is chemically sound regardless of how the computer happens to be looking at it, right? Is that where these equivariant graph neural networks, EGNNs, come into play?
Absolutely critical point, yes. EGNNs are pretty much essential for high-quality 3D generation because they address the fundamental physics and geometry of the system. Think about it. If you design a molecule in the computer, and then you simply rotate your view of it by 90 degrees, it's still the exact same molecule, right? The chemistry hasn't changed. Equivariance is a mathematical property that ensures the model's output, the generated structure, remains consistent or equivariant, even if the input coordinates are rotated, reflected, or translated in 3D space. Without this built-in geometric consistency, the model would mistakenly treat a rotated version of the same molecule as an entirely new, distinct entity. Its predictions about geometry and interactions would be meaningless.
Got it. So EGNNs are like embedding the laws of 3D space directly into the neural network. It's a mathematical guarantee that the blueprint holds true no matter how you turn the molecule model on its side in the computer. So how successful are these combined approaches, diffusion models paired with EGNNs?
The combination has proven incredibly powerful. When you pair diffusion models with equivariant architectures, you see model names like EDM, GCDM, JODO incorporating these ideas. They achieve phenomenal results in terms of structural validity and stability. We're seeing reports where these models achieve over 98.5% EDM stability in complex molecular generation tasks. That means the generated structures are not just geometrically plausible, but chemically stable, which is crucial. This combination, diffusion plus equivariance, is rapidly becoming the sort of gold standard for high-quality, geometrically sound de novo molecular design, especially in 3D.
Okay, we spent a good bit of time on graph structures and 3D geometry. But what about that sequence modeling approach we touched on earlier, the transformer architecture, like in LLMs? How does that fit into the drug design picture?
Transformers definitely have a role to play, leveraging their powerful attention mechanism. This allows them to capture long-range dependencies within a sequence like chemical interactions between atoms that are far apart in the linear string representation, but might be close in 3D space. Older sequence-based models often miss these crucial long-range effects. For instance, models like Chimberta essentially treat molecular design as a sequence-to-sequence translation problem. Often, they work with SMILE strings, those one-dimensional text-based representations of chemical structures. The advantage here is speed and the ability to process vast amounts of existing chemical data, including text from scientific literature, very efficiently.
And the reported efficiency metrics you mentioned for these transformer-based optimization approaches sounded pretty striking.
They really are significant. Studies have shown that these transformer-based optimization methods can significantly improve binding free energy, that's delta dollars, a key measure of how tightly a drug binds to its target, by as much as 2.3 kilocoulomb mol compared to relying solely on traditional, manual medicinal chemistry design cycles. Now, for any medicinal chemist listening, improving binding energy by 2.3 kilocoulomb mol is huge. That's often the difference between a molecule that's just a weak binder and ultimately fails, and a potent lead candidate that actually moves forward into further testing. It can turn a maybe molecule into a definite yes. And almost as importantly, these models have been shown to reduce the typical design cycle times by a remarkable 60%. That's computational power translating directly into compressed R&D timelines and cost savings.
So the progression in complexity seems really clear then. We started with simple 1D strings like SMILES, then moved to 2D graphs showing connectivity, and now the frontier is really these fully resolved 3D geometric structures.
That's exactly right. That is the technological frontier everyone is pushing towards. We move from 1D strings, which are easy to process but lose a lot of information, to 2D connectivity graphs that capture the bonding patterns, and finally, to the full 3D geometric structures. And that 3D conformation is absolutely paramount because ultimately that's what dictates the molecule's physical shape and how it will actually interact and bind with the target protein in the body. Binding is a 3D phenomenon. That's precisely why these cutting-edge models like JODO, the joint 2D and 3D diffusion model, are generating so much excitement. They're designed to jointly learn and generate both the 2D graph, the bonding, and the 3D geometry simultaneously. The goal is to ensure that the computationally generated sequence or graph corresponds directly to a physically feasible and stable 3D structure, maximizing the overall quality and relevance of the final molecular samples for drug development.
Okay, that's a great overview of the toolbox. Let's shift our focus now to how these powerful architectures are actually being put to use in the application that, let's face it, drives the majority of pharmaceuticals sold today, small molecules. We've established we're moving from just passive screening to active generation. And you mentioned we can kind of divide this application into two main types, target agnostic and target-aware design.
That's a good way to categorize it, yeah. Target-agnostic design is essentially about demonstrating the AI's generative capability in the first place. Can it speak chemistry correctly? So the goal here is just to create valid, chemically stable, and unique molecules that look like they could potentially be drugs, but without yet specifying a particular biological target they need to hit. This stage often uses simpler benchmark datasets like QM9, which has small organic molecules, or sometimes more complex drug-like datasets such as GOM drugs. You see models like MEDI achieving really impressive metrics here, like demonstrating 97.5% molecule stability on QM9. It basically proves that AI understands the fundamental rules of chemistry construction.
Right, it's like learning the grammar before writing an essay. But the real therapeutic power, and let's be honest, the commercial value, comes in target-aware design, sometimes called structure-based drug design, or SBDD. This is where the molecule is purpose-built for a specific protein lock, right?
Yeah.
Absolutely. This is where the rubber meets the road for drug discovery. It requires generating molecules that are specifically designed to fit, often very precisely, into a known 3D structure, usually the binding pocket of a specific target protein implicated in a disease. The metrics for success here are naturally much more stringent and application-focused. We rely heavily on predicted binding affinity scores, like the Venus score, where a lower, more negative score indicates tighter, better binding. We also need to consider essential drug-like properties, things like QED, that's quantitative estimate of drug likeness, and SAS score, synthetic accessibility score, which estimates how easy or hard the molecule would be to actually make in a lab. We saw models like DIFF-SBDD, for example, achieving a highly favorable average Venus score of 9.333 on a major benchmark dataset called Crossdocked 2020. That score suggests really promising predicted binding capacity for the generated molecules against their intended targets.
Okay, so the AI can generate promising candidates quickly, but that impressive speed of generation is really only the starting point, isn't it? The other critical half seems to be how AI then fine-tunes those initially generated molecules using sophisticated optimization strategies. This is where we really try to maximize the chance of success further down the very expensive development pipeline.
Indeed. That optimization step is crucial. And one of the most powerful tools we look to, specifically for this stage, is reinforcement learning, or RL. RL essentially treats molecular design like an optimization game. An agent, the AI model, learns to navigate the vast chemical space by taking actions, like adding or modifying atoms or bonds. The model is then rewarded for achieving complex, often multi-objective optimization goals. For example, you might reward it for simultaneously maximizing the predicted binding affinity to the target and minimizing predicted off-target toxicity or improving solubility. It learns to balance these often competing objectives. The release framework was an early, classic example of this, combining RNN generators with predictive networks that guided the generation process towards molecules with better properties.
And the results reported from using this kind of RL optimization approach sound pretty stunning, particularly when you look at actual translational applications for diseases like cancer.
Yeah, the results really do speak volumes about the quality of the designs that can emerge from RL-guided optimization. In one key example, the development of JAK2 inhibitors for oncology using RL-driven optimization dramatically increased the hit rates. That's the percentage of computationally designed molecules that, when actually synthesized and tested, showed potent activity. The hit rate jumped from less than 1%, which is typical for random or traditional screening, all the way up to 10% for the RL-designed batch. That's a tenfold improvement right there. But here's the kicker, the massive business insight, the real so what for drug companies. 85% of those potent RL-designed molecules also exhibited favorable CYP450 inhibition profiles. Compare that to only 45% having favorable profiles in conventional chemical libraries screened for the same target.
Okay, wait, 85% versus 45% on CYP450. That sounds like a huge difference. For the listener, maybe remind us why that specific CYP450 profile is so critically important in drug development.
It's absolutely critical. The CYP450 family of enzymes, they're located mainly in the human liver, and they are responsible for metabolizing the vast majority of drugs that we take. Now, if a new drug strongly inhibits these CYP450 enzymes, it can lead to really dangerous drug interactions when a patient takes it alongside other medications that are also processed by the same enzymes. Levels of other drugs could build up to toxic concentrations. So by designing drug candidates that preemptively have favorable non-inhibitory CYP450 profiles right from the start, the AI is effectively solving a massive, incredibly expensive late-stage failure point before the drug even gets close to human trials. It significantly de-risks the entire development process by building inherently safer, more viable drugs from the ground up. That's a huge potential cost saving.
Okay, so RL helps address safety and efficacy, but what about the practical side? We need to ensure these amazing molecules the AI designs are actually feasible to synthesize in a real chemistry lab, ideally without breaking the bank on complex multi-step procedures.
That's another crucial consideration, and it's where different optimization frameworks come in. Monte Carlo tree search, or MCTS, algorithms used in frameworks like Synthemol become really essential here. You can think of MCTS as being like a sophisticated GPS navigation system specifically designed for chemistry. It doesn't just try to find the best destination, the most potent drug molecule. It uses clever search algorithms to intelligently navigate the vast possibilities of chemical space by specifically prioritizing molecules that lie on plausible synthetic pathways. It looks ahead, considering how the molecule might actually be constructed step by step. It ensures that the estimated chemical yields each step are likely to be favorable, say maybe greater than 80% per step, so that what the AI designs is not just theoretically potent, but also chemically feasible and potentially inexpensive to manufacture at scale. And these MCTS approaches are incredibly efficient too, allowing for the exploration of over 108 BAD, that's 100 million potential candidate molecules per GPU per day, drastically accelerating the exploration while keeping that vital synthetic accessibility in mind.
Mhm. Right, balancing the ideal with the practical. Let's quickly revisit those case studies you mentioned that really illustrate this translational impact in action.
Sure. So back in oncology, those RL-generated JAK2 inhibitor candidates didn't just look good on paper. They showed potent in vivo efficacy when tested. 3 specific candidates demonstrated over 80% tumor growth inhibition in actual xenograft mouse models. That's a strong preclinical signal. Then, moving to CNS drug development central nervous system disorders, the complexity ramps up significantly because molecules generally need to penetrate the blood-brain barrier, the BBB, to be effective. That's a huge historical hurdle. MCTS algorithms were successfully used to generate novel molecules targeting dopamine D2 receptors, relevant for diseases like Parkinson's or schizophrenia. These generated molecules were specifically predicted to be BBB penetrant, showing a log BB score greater than or equal to 0.3. And critically, 90% of these promising CNS candidates also maintained practical synthetic accessibility with a SAS score less than or equal to 4.0, meaning they should be reasonably easy to synthesize. These kinds of results really demonstrate that AI is starting to make headway in conquering fundamental drug development hurdles, like achieving BBB penetration, that have plagued traditional CNS drug discovery efforts for decades. Okay, so if small molecules represent one level of complexity, then biologics, these large therapeutic molecules like proteins, peptides, and antibodies, are, let's say, exponentially more complex. Their biological function depends entirely on folding into a very precise, intricate, and often dynamic 3D structure. And that structure is dictated by a long sequence, sometimes hundreds or even thousands of amino acids chained together. This relationship between the 1D sequence and the 3D structure is often called the folding problem, and it's been a grand challenge in biology for 50 years.
Right. And the absolute foundation of this entire field, the breakthrough that really changed the game, was, of course, protein structure prediction, the famous AlphaFold 2.
AlphaFold 2 remains a true landmark achievement, no question. It achieved near experimental accuracy in predicting protein structure from sequence alone specifically, an average of 3.30 Angstrom RMSD across many test proteins. It completely dominated the CISP assessment, the critical competition in the field, and effectively solved the long-standing protein folding problem. At least for single protein chains, it was revolutionary. However, for integrating this into a high-throughput drug discovery pipeline, prediction speed also becomes absolutely crucial. And that's where RosettaFold came in as a really vital parallel development. RosettaFold demonstrated comparable accuracy to AlphaFold 2 on many targets, but with significantly improved speed. It could generate structure predictions or samples in around 10 minutes, which was roughly 100 times faster than early versions of AlphaFold. That speed is what translates directly into scalability for things like virtual screening or designing new proteins.
Okay, predicting structure from sequence is amazing. But I find this next concept even more mind-boggling. Once you know the structure you want, the challenge becomes generating a sequence that will reliably fold into that specific structure.
Yeah.
It feels like we're asking the AI to literally reverse engineer nature itself. This is the inverse folding problem, right?
It really is like reverse engineering biology at its core. You start with a fixed desired protein structure, maybe a completely novel scaffold designed computationally, often just represented as the backbone atoms, and the task is to generate the optimal corresponding sequence of amino acids that will naturally fold up into that exact target shape and be stable. The state-of-the-art model leading the charge here is called Protein MPNN. This model uses a sophisticated graph-based neural network architecture specifically designed for sequence optimization given a structural context, and its success metrics are genuinely astounding. On benchmark tests, it achieved around 52.4% sequence recovery, meaning it correctly predicts over half the amino acids in a known protein just from its backbone structure. Even more impressively, in practical tests, it successfully rescued 12 out of 15 13 previously failed computational protein designs generated by older methods like Rosetta. It did this by finding better sequences that properly optimize the interactions between residues, turning prior computational failures into experimentally validated successes. That's huge.
We saw how diffusion models really started to dominate the small molecule design space due to their stability and quality. Are they making similar waves in protein design as well?
Oh, absolutely. Diffusion models are proving just as impactful for proteins. A leading example here is RF diffusion, which very cleverly repurposed the architecture of Rose decafold, the fast structure predictor, and turned it into a generative diffusion model. RF diffusion works by iteratively refining protein backbone conformations through those powerful denoising steps we discussed earlier. It starts with noise and gradually sculpts it into a coherent protein structure. It's achieving sub-angstrom precision in generating entirely new protein backbones and scaffold shapes that have potentially never existed before in nature. This isn't just prediction anymore. This is active de novo protein design at the most fundamental atomic level.
Okay, let's pivot slightly to the highly specialized but hugely important world of antibody engineering. Antibodies are these large, Y-shaped proteins that our immune system uses, and we engineer them as therapeutics to bind very, very specifically to targets like viruses or cancer cells. What's the major computational hurdle in designing a new antibody?
The primary challenge, the real bottleneck in computational antibody design, lies in accurately predicting the 3D structure of the loops at the tips of the Y-arms. These are called the complementarity determining regions, or CDRs. These CDR loops are what actually make contact with the target, the antigen, and among them, the CDRH3 loop is notoriously difficult. It's the most variable in both its amino acid sequence and its length across different antibodies. The precise structure of that CDRH3 loop pretty much dictates the antibody's binding specificity and affinity. It's the absolute key to locking onto the intended target and ignoring everything else. Historically, these loops were so diverse and flexible, they were almost impossible to model accurately with computation.
Okay, so a highly variable critical loop structure. How are the modern foundation models, especially the language model types, tackling this specific CDRH3 challenge, particularly with speed in mind?
This is where models like IgFold represent a major breakthrough, really leveraging the power of sheer data scale, similar to LLMs. IgFold uses language model principles, but it was pre-trained on an absolutely enormous data set of 558 million known antibody sequences. That vast amount of data allows it to learn the subtle patterns and constraints governing antibody structure, including the tricky CDR loops. As a result, IgFold achieves a really significant improvement in accuracy, getting the CDRH3 loop structure prediction down to an average RMSD of just 2.38 angstroms. And crucially, it does this incredibly fast in less than 25 seconds per antibody. That speed is utterly transformational for the field. It means researchers can now realistically construct and screen massive virtual libraries containing millions or even billions of potential antibody designs in a time frame that was completely unimaginable just 5 or so years ago.
That speed and accuracy sound incredible, and we've seen continued advancements since then, particularly with the latest AlphaFold 3, or AF3, which came out recently. The report said it achieved an even better median CDRH3 RMSD, something like 2.04 angstroms, for unbound antibodies. That sounds like we're approaching near-perfect accuracy for that critical loop. So why is antibody design, especially predicting how it binds, still considered such a fundamental computational challenge if we can model the pieces so well?
That's where you have to introduce a really critical nuance, a major challenge that remains. Accuracy on the individual components, like the antibody alone, is necessary but definitely not sufficient. The entire purpose of a therapeutic antibody is to bind tightly and specifically to its target antigen, usually another protein. So what you really need to predict accurately is the structure of the entire antibody-antigen complex when they are bound together. And this is where even the latest models, like AF3, still struggle significantly. Despite its impressive precision on single, unbound proteins or antibody structures, reports indicate it still fails to predict the correct binding mode, the correct antibody-antigen interface, in approximately 60% of cases, especially when relying on standard prediction settings like single computational seeds.
Wait a minute, 60% failure rate in predicting the actual binding complex. Even if AF3 gets the CDR loop structure down to, say, 2 angstroms accuracy when it's floating alone, if it fails to predict how it actually connects to the target correctly more often than not, doesn't that kind of make the individual accuracy almost irrelevant for practical drug design? The complex is the only thing that really matters for therapeutic effect, right?
You've hit on a profoundly important point, and it raises deep questions about the current limitations of even our best foundation models in biology. Predicting the interface, the precise way two complex, flexible molecular surfaces will interact, dock, and settle into a stable, low-energy bound state that turns out to be a significantly harder computational problem than predicting the structure of a single molecule in isolation. That 60% failure rate for predicting the complex starkly highlights that while FMs like AlphaFold have essentially solved the static protein folding problem for single chains, they have not yet fully solved the dynamic protein binding problem, or what's sometimes called the docking problem. Accurately modeling that interface, especially for flexible partners like antibodies, remains the next major computational frontier. We absolutely must conquer that challenge to fully leverage these incredibly precise generative tools for designing truly effective biologics like antibodies. Predicting the parts is great, predicting the interaction is the goal.
Okay, so the revolution clearly goes way beyond just inventing molecules in the lab, whether small or large. It seems foundation models are also starting to streamline the back half of the incredibly long and expensive biopharma pipeline. We're talking clinical trials, diagnostics, operations, which is frankly where the bulk of that enormous $2.8 billion average cost is actually incurred. This sounds like where AI moves from pure chemistry into logistics, data analysis, and even text processing.
Absolutely. And this is where the natural language processing capabilities of foundation models, the very same kinds of capabilities that power the LLMs we interact with daily, really start to shine, but applied to clinical informatics and administrative medicine.
Okay, such as maybe trying to encode the, let's face it, often chaotic mess of complex patient records into something actually useful for analysis.
Exactly that. Models like BERT, which, as the name suggests, is a BERT-like transformer adapted for healthcare records, are being applied directly to patient data. They learn to encode the highly unstructured, often text-heavy information found within electronic health records, or EHRs, into standardized, usable numerical representations. These representations can then be used for really complex downstream tasks, like finding patients with similar disease trajectories, improving cohort selection for clinical trials, or even performing early risk prediction for things like hospital readmissions. This can significantly speed up trial recruitment and parent stratification, which are major bottlenecks. And then on the diagnostic support and medical knowledge front, the MedPalM model, developed by Google, was a truly historic milestone. It became the first AI model to successfully pass the U.S. Medical Licensing Examination, the USMLE. Now, passing a licensing exam doesn't make it a doctor, obviously, but it confirms the incredible depth of synthesized medical knowledge these foundation models can now possess and reason over.
Okay, that's processing existing knowledge and data. Well, let's talk about predicting the future, specifically predictive clinical trials. You mentioned traditional human trials are incredibly high risk, costing maybe $2 billion for later phases, with a final approval rate stubbornly hovering around a dismal 15%. This sounds like a perfect target application for AI to try and reduce that massive financial risk exposure for companies.
It really is perhaps the ultimate application of predictive modeling to de-risk the entire drug development pipeline at its most expensive stage. AI models are being developed, like one called the Hierarchical Interaction Network, or HINT, which are specifically pre-trained on vast amounts of multimodal drug data. That means they learn from integrating chemical structure information, known toxicity data from previous experiments, existing clinical trial protocol data, outcomes from past trials, and even real-world evidence from patient registries. The goal is to use all this integrated data to simulate the complex interplay of factors within a clinical trial and actually predict the likely outcome, success or failure, and maybe even specific adverse events before the incredibly expensive large-scale human trials even begin.
Okay, predicting outcomes is one thing, but how does a model like Hint specifically help chip away at that massive, say, $2 billion average cost for bringing a drug to market?
It chips away at the cost primarily by allowing researchers and pharmaceutical companies to simulate hundreds, maybe thousands, of potential trial designs and, crucially, predict potential failure modes entirely in silico on the computer. So, for example, if the HINT model predicts with very high probability that a particular drug candidate is likely to fail its phase 2 trial due to, say, unacceptable liver toxicity signals based on its structure and preclinical data, the company can then make a much more informed decision. They could potentially terminate that specific trial early, or perhaps go back and try to re-engineer the drug candidate to mitigate that toxicity risk before spending hundreds of millions of dollars on patient recruitment, hospital site administration, monitoring, and data collection for a trial that was likely doomed from the start. This ability to simulate failure to get an early warning signal is a massive potential cost-saving measure. It could dramatically shift the financial calculus and risk profile of drug development if these predictions become reliable enough.
Right, avoiding costly failures is key. Finally, there's another fascinating application area you mentioned, seemingly quite different, materials science and engineering, specifically optimizing drug delivery systems. How does AI tackle something as complex as designing, say, a nanoparticle NP to carry the drug?
Yeah, this is a really interesting intersection. Nanoparticle delivery systems, especially things like lipid nanoparticles, LNPs, which became famous for delivering the mRNA vaccines, are themselves highly complex engineered materials. Their composition and structure are critical. AI becomes crucial here because often you need to optimize not just the drug molecule itself, but how it's packaged, protected, and delivered effectively to the specific site of action in the body while avoiding accumulation elsewhere. AI, particularly deep learning, is being used to predict the complex behavior of these nanoparticles once they're inside the body. For instance, deep neural networks have been trained on large databases compiled from studies involving tumor models. And these DNNs successfully predicted the degree of nanoparticle accumulation in different types of tumors with considerable accuracy. One study reported an R-squared value of 0.70, which is quite good predictive power.
Okay, 0.70 R-squared is solid for predicting accumulation, but how do they make those predictions even smarter by integrating them with, like, the actual mechanics of how things move around the human body?
That's the next level of sophistication, combining the data-driven AI approach with mechanistic biological modeling. Hybrid AI PBPK models are emerging as a powerful tool here. PBPK stands for physiologically based pharmacokinetic modeling. These are traditional models that simulate how a substance moves through the body based on known physiological parameters, blood flow rates, organ volumes, metabolic enzyme activities, etc. By combining the AI's ability to learn complex patterns from data with the PBPK model's representation of physiological processes, researchers have further enhanced the prediction accuracy for things like NP delivery. One study achieved an R-squared value of 0.83 for predicting tumor delivery efficiency using such a hybrid approach. Now, what this really means in practice is that researchers can simulate the effects of subtle modifications to the nanoparticle design, maybe changing the specific lipids used in an LNP formulation, or altering the particle size and predict how those changes will affect organ-specific exposure patterns and delivery efficiency, all computationally. This significantly reduces the need for resource-intensive, time-consuming, and often ethically complex animal testing for every single design iteration. It dramatically accelerates the rational design process for creating highly targeted and effective drug delivery systems. So, okay, we've painted a picture of these really powerful generative models already driving significant efficiency gains across the board, but we absolutely must be clear-eyed about the gaps that still exist. There are persistent challenges in bridging the gap between computational prediction and messy experimental reality. The work is definitely not complete.
Right. So where do these impressive models typically start to struggle? When do they leave the sort of controlled environment of a clean benchmark data set and hit the complexities of the real world?
Scalability and handling real-world complexity remain the main hurdles, I'd say.
Yeah.
While many models perform excellently, sometimes achieving near-perfect scores on simpler, well-behaved benchmark datasets like QM9, they often start to struggle when faced with the sheer complexity, the increased size, and particularly the conformational flexibility, how much a molecule can wiggle and change shape of actual drug-like molecules found in more realistic datasets like GUM drugs. It's kind of the difference between designing a simple, rigid Lego brick versus engineering a highly complex, flexible machine with many moving parts. The real drug targets and candidates are more like the complex machine. Similarly, in protein design, we still face persistent challenges in generating highly accurate structures for larger and more complex proteins, especially those maybe over 128 amino acid residues or proteins that have complicated interactions, like those embedded within cell membranes. Size and complexity still push the boundaries.
And if every research group, every company, is developing their own cutting-edge model, maybe their own customized version of a diffusion model or a specific GNN, how do we, as a field, objectively compare performance? How do we know who's actually making progress?
That leads directly to another really critical issue facing the field right now: the need for better standardization, especially in benchmarking. Generative tasks, particularly in protein design, things like designing completely new protein scaffolds from scratch or engineering specific binding interfaces, currently suffer quite badly from a lack of standardized, widely accepted benchmarking procedures and datasets. What happens now is that nearly every published paper reports its own success metrics, often calculated in slightly different ways, usually on its own bespoke, internally curated dataset. This makes it incredibly difficult, sometimes impossible, to objectively compare the quality and performance of different models across different studies. Is Model A truly better than Model B, or do they just use an easier test set? Establishing common, rigorous, and widely shared benchmarks, something akin to the CASP assessment for protein structure prediction, is absolutely essential for accelerating future progress. We need a level playing field to really determine which new methods and architectures are truly leading the field forward.
Okay, standardization is key for progress. Now let's bring it back to the huge industry implications we touched on earlier. The technological progress is clearly driving monumental investment, maybe even reshaping market caps, as we discussed with the NVIDIA comparison. What's the final trajectory here? What's the big picture vision that makes this level of investment seem perhaps inevitable?
I think the future trajectory, the big vision that's driving a lot of this, is the concept of the truly integrated closed-loop system, essentially moving towards a kind of automated AI-driven drug discovery factory. We're moving rapidly towards much tighter integration between the in silico design phase, the AI generating predictions and hypotheses, and immediate, ideally automated experimental validation of those predictions, closing the loop between computation and experiment faster and faster.
That sounds incredibly ambitious, an automated drug discovery factory. What does this closed-loop system actually look like in practice on the ground?
Well, you can imagine it combining the sophisticated AI algorithms we've discussed with advanced, automated, high-throughput synthesis platforms, robots that can actually make the molecules the AI designs. And then, critically, integrating that synthesis with comprehensive, high-content in vitro testing platforms. Increasingly, people are looking towards technologies like organ-on-a-chip, or OAC platforms. These OAC systems use microfluidics to create miniature functional models of human organs, like a tiny liver on a chip or lung on a chip. They can offer significant advantages over traditional cell cultures or even animal models by providing more physiologically relevant human-specific data on efficacy and toxicity early on.
Wow. Okay, so the AI designs a potential drug molecule. An automated robotic platform synthesizes that exact molecule, and then an organ-on-a-chip platform immediately tests its efficacy and toxicity in a miniature but functionally relevant human-like biological environment. Is that the idea?
That's exactly the vision. And the closed-loop part is crucial. If the newly synthesized molecule fails the OOC test, maybe it shows unexpected toxicity, or it isn't as effective as predicted, that failure data, the experimental results, are immediately fed back to the AI model. The model can then be retrained or fine-tuned using this new real-world data, allowing it to learn from its mistakes and systematically analyze the reasons for failure. This continuous feedback loop promises to dramatically accelerate the entire design, make, test, analyze cycle, potentially shrinking it from years down to weeks or even days for certain iterations. This convergence of technologies, high-precision computation meeting high-throughput automation, and advanced biological models, like OSCs, this is what holds the potential to truly drive down the astronomical costs and crippling failure rates of traditional drug development. I think it's highly likely that the next decade will see these AI-designed, rapidly validated therapeutics becoming absolutely central to the future of precision medicine, where treatments are tailored atom by atom for specific disease mechanisms and potentially even individual patient populations.
It's really quite something to consider. That entire pipeline, literally from predicting the precise atomic coordinates for a novel protein binder all the way through to designing and streamlining a multi-billion-dollar clinical trial, all being fundamentally reshaped, maybe revolutionized by these foundation models. The scope is just enormous. It feels genuinely historic.
It really does feel historic. I think the most fundamental shift, the thing to really internalize, is that we're moving from an era of passively understanding biology, observing, analyzing, and characterizing what nature already provides us, to an era of actively designing biology. And that active design capability is being driven by these computational models achieving, in some cases, atomic-level precision in their predictions and generations. This represents a massive acceleration, a scientific force multiplier that promises to fundamentally enhance our ability to tackle complex diseases that have evaded cures for so long.
So, as you, our listener, go back to your own work or just ponder these ideas, here's a final provocative thought we want to leave you with to mull over. Considering the almost unbelievable success of predictive models like AlphaFold 2 in solving the protein folding problem, and now the incredible high-speed generation capabilities we're seeing from new diffusion models like RF diffusion for creating entirely novel structures, how soon might it be until we start relying on AI's structural predictions and designs, not just as a helpful computational starting point for experiments, but perhaps as the final gold standard truth of a designed therapeutic molecule? Could we reach a point where the confidence in silico design is so high that it significantly alters or even precedes the need for extensive traditional experimental validation? That's something to think about.